Hello, we are going to present you our genetic engineering final project named Obtainment of nitrate reductase via the overexpression of NIA gene in Micromonas pusilla for its use in seawater denitrogenation. Taking into account the current nitrogenation and acidification problem in the ocean, we decide to create a recombinant vector for the overexpression of nitrate reductase to help with the denitrification. In our project, NIA gene from algae was taken and enhanced with a special promoter, both inserted into a GIP vector. The recombinant vector was used to transform Saccharomyces cerevisiae cells to obtain the metabolites containing the desired enzyme for their use in ocean denitrogenation.
Well, so the UN Sustainable Development Goal number 14 is to conserve and sustainably use the oceans, seas, and marine resources for sustainable development. It also says that marine biodiversity is essential for the health of our planet and its population. Nitrogen is essential for many biological functions because most plants, algae, and microbes require biologically available or fixed forms of nitrogen. Nitrogen is one of the worst pollutants in the world because it damages the oceans, it is the cause of proliferation of harmful algae, it also contributes to eutrophication and dead zones in the oceans, and it has also some other negative side effects. Nitrate has proven to be toxic for living organisms such as humans, plants, and animals because it affects its ability to transport oxygen. This Sustainable Development Goal number 14 of the UN can be solved thanks to genetic engineering via bioremediations, according to the EPA. We plan on using the algae's bioremediation functions with its capacity to absorb nitrogen, heavy metals, and other toxicants in aquatic environments so that we can contribute to the process of denitrification in the ocean. Okay, so the nitrogen cycle has two pathways, nitrification and denitrification. Nitrification is a process in which ammonium is converted into nitrate by oxidation with the help of soil bacteria or bacteria. Denitrification is important for is the reduction of the nitrate to nitrogen and is done with the help of bacteria that produce nitrate reductase enzymes. Denitrification, as I was about to say, is important for fixed nitrogen budgets in the oceans since it is a primary loss mechanism for nitrogen when it is turned into its gaseous form and released to the atmosphere. Nitrate can easily dissolve in water, and once there, it is hard to remove.
This project seeks to make the most out of algae accumulation while solving the excess of nitrogen in seawater through denitrification. This will lead the ocean and marine resources preservation. Nitrate reductase plays a huge part in denitrification, and algae can produce this enzyme. The specific objective of this project is to generate a recombinant vector which might cause the overexpression of nitrate reductase. To avoid the introduction of an exogenous species into the ecosystem, useful metabolites containing the nitrate reductase will be purified from the transformed organism.
Several organisms, such as algae, are helpful with the process of denitrification due to the production of nitrate reductase. This is the case of Micromonas pusilla, a green unicellular marine algae. The NIA gene found in this algae is responsible for codifying for the enzyme. The gene size is approximately 1.6 kilobases. The sequence was obtained from MCBI. This is the gene that will be overexpressed in the recombinant vector. An enhancer-like promoter for the NIA gene was found in Arabidopsis thaliana. It is called NIA1 promoter and its approximate size is 1.8 kilobases. This promoter might be right next to the NIA gene or even several bases away from it. Since it induces translation, when added to the recombinant vector, overexpression up to 4-fold might be achieved. A GIP vector was selected for its integration into Saccharomyces cerevisiae's DNA. The chosen one is the GIP lag 204 TKC E2 crimson HDEL. It contains an ampicillin resistance gene, which will act as a selectable marker. This vector expresses E2 crimson, but this gene will be cut out and replaced with the promoter gene insert.
For this to be achieved, several steps need to be taken. First, an RNA extraction from Micromonas pusilla is done, followed by an RT-PCR for obtaining the NIA gene and a verification with electrophoresis. DNA from Arabidopsis thaliana is extracted using the CTAB method. Following previous studies, a restriction digestion with enzymes HIND3 and PST1 will allow us to get the NIA1 promoter. A second electrophoresis followed by a gel extraction and a normal PCR for amplification of the promoter are done. The first ligation will be done for joining the promoter and gene, and for these adapters are needed. The vector obtained from the Glick laboratory is digested with restriction enzymes X-BAL and E-cori for cleaning cutting E2 crimson out. The procedure is verified with a third electrophoresis and gel extraction. The final ligation is done with the insert of the vector, once again with the help of adapters. The recombinant vector is used to transform yeast cells with the lithium acetate method. Yeast cells are grown and reproduced in YPAD medium. The colony selection is done in SE media with ampicillin.
Here is the recombinant vector that will be obtained following the methodology previously described. The model was made in Snapgene. The purple part represents the NIA1 promoter, which will enhance an induced translation of the NIA gene shown in blue. Both of them are inserted into the GIP vector in the ideal position for undergoing translation. A tree restriction enzyme method will be done to verify its orientation and integration using a EcoR1 and HIN for carrying HIN3 for carrying the insert from the vector, along with the APA1 which will cut between the insert.
The transformed yeast will be grown in a suspension culture. The metabolites in the liquid will be then purified using a high-performance affinity chromatography with specific antibodies for enzyme recovery. It will then be tested in seawater samples with a measured nitrogen concentration to monitor their uptake during an established period of time. It will be compared with natural algae and wild yeast applied to seawater, and also an untreated sample as control. Each sample will be done in triplicate. It is expected that the transformed yeast cells overproduce nitrate reductase and that their nitrogen uptake has a higher yield.
The produced transformed yeast cells, if proven to optimize the denitrification process in seawater, can be further studied to determine the effect of their use in seawater. It is suggested for a matrix or bioreactor to be used when applied in the natural environment to avoid any possible residual contamination. Inducing the production of nitrate reductase, like done in this project, might be the first step towards the reduction of ocean pollution and acidification caused by excessive nitrogenation. These are our references. Thank you for watching.